who has been a frequent and fervent contributor to the Quantitative Systems Biology Center here, attendee for a number of years. He's a fifth-year grad student in physics in Bill Holmes's lab, and really looking forward to hearing what he has to say today.
And a real quick note for our attendees, please feel free to drop in questions in the chat box or on the Q&A session, and then we will answer those kind of in brief pauses as John gives his talk. Thanks.
Cool, so get started.
Let's do it.
All right. Sharing my screen. Are my slides visible?
Yes, looks good.
Cool. So welcome to the seminar. I guess the second official seminar of this semester series. I'm John Vastola, a fifth-year graduate student here at Vanderbilt in the Department of Physics and Astronomy. I'm going to be talking to you about some theoretical work that I did with some collaborators at Caltech, in particular Gennady Gorin, top-notch computational biologist in the lab of Lior Pachter. So first, let's go ahead and set the stage. We're going to be talking about transcription, and in particular, splicing dynamics. At a very high level, transcription is more complicated than this cartoon, but you can think of it as you have a DNA template, you use that to construct some RNA, then you process that RNA, for example, by taking out pieces of it called introns, and then you use that RNA for something, for example, to make proteins or to regulate genes, whatever. So this is a thing that exists in the natural world, and we can ask questions about it. One easy to ask, but kind of unconstrained question is, how does transcription work? Another, probably very equally hard or harder question is, can we use what we know about transcription to predict the future? Many of you may be familiar with RNA velocity, kind of a new trendy method for predicting single cell trajectories. And while it has some technical issues with it, I think the general principle is interesting and probably can be carried a lot further, which is that if you measure the number of unspliced RNA in a single cell and the number of spliced RNA in a single cell, then in principle, knowing the number of unspliced RNA right now tells you something about the number of spliced RNA in the future, since one gets converted into the other. And there's ideas about how to use that maybe to study development, or maybe big pie in the sky to control sulfate, stuff like that. So in principle, we could use transcription to understand things like this. What's kind of, first we have to take a step back and say, those are really hard questions, so let's make them more specific and ideally quantitative so we can see how well we're doing. So things like, how long does it typically take to take out a piece of RNA, to splice out an intron? In what order are these introns removed? How frequently does an actively transcribing gene produce RNA? How many gene states and splicing steps are typical? How predictive really are unspliced RNA counts of spliced RNA counts? What's kind of amazing is that questions like this, there's some hope of actually answering them with a lot of recent single cell data. For example, here's a picture, part of a figure from a PNAS paper from just 2 years ago by Brian Munsky and his collaborators, two of which, Huadong Li and Gregor Neuert, are from here at Vanderbilt. And what they did is they made single RNA molecules fluoresce, and they can count the little bright dots to count in living cells how many RNA there are, both in the cytoplasm and in the nucleus. And then you can use those counts to make interesting quantitative models of what's happening. And this is really just the beginning. There's other technologies, like a couple that my collaborator Gennady Gorin has told me about, like nanopore direct RNA sequencing and Tron-seqfish, that have some hope of maybe not just counting RNA, but being able to identify how many RNA have 1 intron currently removed or 2 introns currently removed or stuff like that. There's a lot of technical issues with these things, but there's real hope of having the data to answer these kind of fantastically difficult and high resolution questions. And so if you have these questions, how long does this take? How frequently does this happen? And so on. What you really want is some sort of quantitative, interpretable model. In other words, generally speaking, if you can fit your data, you fit a line or a curve or whatever, then I don't know, who knows what that says? Maybe you can predict things, maybe not. We want models that say this curve fits, and so here's what that tells us about what's actually going on. At a high level, you might say, in order to fit the single cell data, we need 1, a model, and 2, a way to extract model predictions. So the central question of the talk is, how do we efficiently extract model predictions from mechanistic models of RNA splicing dynamics? There's a lot of models you could pick. I think probably the kind that most people here would be familiar with are ODE models. And these are, simple, and maybe they'll give you a coarse idea of what's going on, but they only really apply, and don't always apply in this case, when you have lots and lots of mRNA in a cell. But that's not always true. A lot of times you have 10 or 3 or something, and whether the number is 10 or 3 could really impact qualitatively the cell fate. There's other kinds of models, SDE models, Boolean network models, Markov models. Each of these has its own time and place. But for our purposes, what we really need in order to understand real counts data is we need some model where molecules are modeled as having discrete numbers, 0, 1, 2, 3, instead of concentrations like 1.7 nanomolar or something, where the dynamics are treated as stochastic, since when you get to the low copy number regime, it turns out that you're really affected by things like the randomness associated with molecular diffusion and binding and unbinding and so on. So we need discreteness, we need to be stochastic. Probably don't need so much information about spatial positions. Like I don't need to know that the mRNA is right here instead of right there. Maybe we need compartmental information, like it's in the nucleus versus cytoplasm. But with all these requirements, it turns out that the standard theoretical framework for this is something called the chemical master equation. And models in the framework of the chemical master equation involve 3 things. If you think of it like a play, one thing is the actors. For example, in this little picture, unspliced RNA at the top, spliced RNA at the bottom. And then there's the things that happen to the actors, the action. And the things that can happen are unspliced RNA can be produced, unspliced RNA can be spliced to become spliced RNA, and spliced RNA can be degraded. And then you have numbers, parameters, or more technically you need propensity functions, which quantify how often each of those things happens. And if you can do those things, very easy to write down a model, although solving it is a different story. It's worth keeping in mind that the way that the CME views the world, discrete and stochastic, means that you can't exactly predict the future state of a cell given its current state, which you could with something like an ODE model. Instead, really the fundamental thing is to think about probabilities. And so the CME answers the following question. Given what the probability distribution for mRNA counts or protein counts or whatever looks like right now, how will it look sometime in the future, 5 minutes from now or an hour from now or whatever? That's the question that it answers. And naively, you might say, well, this is a pain because imagine it looks like this right now versus that right now. Maybe since the starting kind of distributions are very different, they'll evolve into completely different things and we'll really have no hope of understanding what's going on. But a nice feature of these kinds of theoretical models is that they tend to converge to a unique probability distribution. So here in this little picture, you can see just whatever random initial distributions, if you wait long enough, which on experimental timescales you are, it looks like the same thing. So theoretically, it's very nice. And it also turns out to be true that you can get data for something like this, and people have, like in that PNAS paper I mentioned. Okay, so CME models, and we want to extract the steady state probability distribution to compare with experiments and fit parameters and stuff. So how do we calculate PSS? Usually you do it via stochastic simulations, which you can see a bunch of random ones shown here. That's the normal way, but that's not the only way. And that leads me to the central slogan of the talk, where if you don't pay attention to anything else, I think at least these are two important takeaways. One, simulations are not the only way. And two, they may not always be the best way. What do I mean by that? Well, there's kind of a trade-off between how difficult it is to implement your method for extracting predictions for your model versus how fast that runs. And that can become very important when you're doing parameter inference, and you need to evaluate model predictions on like hundreds or thousands of models. So for example, this is not real data. This is just something I made up to convey the idea. For solving the CME, I think you could maybe divide the kinds of things you have available into 3 different classes. One of them is simulation-based methods. The next one is like numerical solvers of the CME, which solve in kind of a different way, not involving simulations. And then the other one is just having a math formula that you can write down on paper. Simulation methods are easy to program, and you can like take the same code and make it work for a wide variety of problems, but they're really, really slow. And so again, if you're trying to try 100,000 parameter sets and each one takes 30 seconds, you can have a tough time. The second group in blue, you might imagine stuff like finite state projection. Don't worry about what that does, but it's faster and it takes longer to sit down and actually work out for a specific problem, the code that'll give that to you. And then the best possible thing, the thing that would be the fastest, are these math formulas, but they may or may not exist even for the problem that you're working on. And generally it takes a lot of work in order to find them. Once you have them, it's great, but finding them is hard and usually not possible. For comparison though, for a really simple problem where you just have mRNA that produces, gets produced and gets degraded, you can try 3 examples, one example of each method on this problem to find the steady state probability, and they all work. They're all correct. That's shown here. But the amount of time it takes for each one and the scaling of that time as a function of how big and complicated your system is very, very different. So the math formula is the thing at the very bottom. And no matter how big and complicated your system is, at least in this simple example, it takes about the same amount of time to spit out an answer. For the finite state projection, you can see as the system gets bigger, it takes longer and longer. And the stochastic simulations, it's even worse. So for example, if you have a typical molecule number in your system of like 500, in this case, it takes you 30 seconds per parameter set to make a prediction. And again, if you want to try 100,000 parameter sets to see what fits your data, that's not good. But what do we mean by analytic solutions? Let me say more about that. The chemical master equation, as a math thing, it is an equation. That's the equation. And you're looking for the probability distribution as a function of time and of all your possible states in your system, given some specified initial data, which, as I said, in practice doesn't matter that much. That's the formal math equation. More concretely, in the simple example of an mRNA that gets produced and degraded, it looks something like that. And even though that is solvable, in general, it's very, 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 very hard to solve it exactly, because what it amounts to is trying to solve a system of infinitely many coupled ODEs. So you need to find P0 as a function of time, P1 as a function of time, P2 as a function of time, and so on and so on and so on. And each one affects how the other ones change. At the end of the day, what you really want is you want formulas like the thing at the bottom, which is a Poisson distribution, the steady state probability for this simple birth-death process. It's not always possible. And let me say 2 more slides and then maybe we can stop for questions. You can ask, when is it possible to write down analytic solutions? The short answer is almost never. Probably the biggest class of models where you can write down analytic solutions are so-called monomolecular reaction networks, where basically a reaction list has to consist of some combination of production, degradation, and first-order conversion, a reaction of the form A goes to B. Our splicing model that I'll be talking about is an example of that. There's other simple problems that you can solve, but they're really simple, so maybe not that useful in practice. And then you can say some formal mathematical things about them. For example, I have a paper under review in the Journal of Mathematical Biology, giving a formal solution for arbitrary combinations of 0 and first order reactions, but it's the kind of thing where it's like, it's cool that you can do it at all, but it's not the kind of thing that you could put on a computer or at this point really usefully apply. Meanwhile, what problems are not known to have exact analytic solutions you can write down on paper? Something as simple as Michaelis-Menten kinetics, where you have something that binds and unbinds. Almost anything involving proteins. Generally, anything more complicated than monomolecular, which is kind of a problem. And even worse, generally speaking, most of the CMEs that we know how to solve don't include bursting. Bursting is this biological feature that turns out to be important for understanding transcription and splicing dynamics, where you imagine a gene, instead of just producing RNA at the same rate all the time, maybe it has different states that it switches between. Usually these are called like on state and off state, or maybe there's more than 2. And so you end up with, if you look at the number of mRNA over time in a single cell, is maybe some picture like on the right, where there's like no mRNA, and then the gene turns on and it jumps up, and then the gene turns off and it goes down, and then the gene turns on and it jumps up again, and then it goes down, and so on. And it turns out mathematically, trying to solve the CME for situations like this is very, very hard, heuristically, because you're trying to couple a discrete switching process, on, off, on, off, on, off, to something that's very different, like almost like a random walk on molecule numbers, going from 0 to 1 to 2 to 1 to 2 to 3, stuff like that. So I'm going to tell you about a way to solve this problem, and then what that can mean in practice and how that might be useful. But I guess now's a good time to stop for questions.
All right, we don't have anything in the chat box so far, but if anybody has a question, feel free to raise your hand using the controls at the bottom, and you can speak if that would be easier. If not, the floor is yours, John.
I'm gonna wait a second just because the next stuff is might go on for a bit.
All right, I'll ask a question. So tell us a little bit about the motivation for this, because it's not just about time, right? It looks like you feel there is a more profound understanding when there are analytical solutions. Is that?
Well, I mean, it depends on what you care about. I think it's cool and intellectually satisfying to be able to look at these complicated problems and write down an answer on paper. But on the other hand, maybe a computational biologist or an experimentalist doesn't care. And that's fine. Everybody has their own thing that they care about. So for them, what may be more important is, can I help you fit the data? And if it takes a really long time in practice, maybe you just can't do that. So.
So yeah, I have a quick question for John. I'm assuming you might be getting into it, but one of the things that struck me was the possibility of combining or making a hybrid approach where you'd have some analytical solutions where you can define the mathematical formula, but in others you can do simulations. I have no idea how you would couple those things, but is that something that you've considered or are working on?
It's a good thought. It's not something that I honestly thought about recently, but I suspect probably if you want to really get the maximum, I don't know, power out of these kinds of ideas, you probably need to do something like that. Because like I said, it's really rare that you can solve things at all. So any step forward is a step forward. Maybe it'd still be really good to kind of like solve part of it and then combine that with some simulations or something else. I think I will think about that more now that you said that.
Excellent.
There's something in the chat. Jason, sorry.
So we have a question from Jason Hughes. Thanks, Vito. Can this be extended to non-Markovian systems where you cannot necessarily assume the system is memoryless?
That is an excellent question. I do not know, but I will say that there's kind of a get out of jail free card in the following sense. Usually if you have a non-Markovian system, you can make it more complicated a little bit and then it becomes Markovian. So then maybe you could do that. And then the other thing to say is that generally speaking, you can make some arguments about the physics of molecules binding and unbinding and stuff like that and say the Markovian assumption is pretty good. But it is kind of a difficult thing to consider. Should I continue?
Yeah, I think we're good.
Cool. So the next section of the talk is going to be the meat of it. I'm going to get into some of the technical details, but let me remind you, the purpose is to say, okay, it's really hard to solve CMEs that include this bursting feature where genes turn on and off. So I'm going to talk about a way to do that and illustrate it with a very specific example. And then talk about what you can use it for. So the specific kind of system that we're thinking about is what is sometimes called the telegraph model, but for various reasons, I'm going to be calling birth, death, plus gene switching. And here's a list of the things that can happen. You have a gene that switches between two states. You can call one on, one off, but the production rates could be whatever. Depending on the gene state, you produce RNA at a different rate, and then the RNA can degrade. As I pointed out before, you get this kind of bursting behavior in many cases, which you can see on the bottom right. And then as far as the math goes, the actual equation you have to solve is the thing on the bottom left. It looks kind of complicated. And it turns out, if you think it looks complicated, it's even more complicated than you expect it to be. So Gennady suggested this problem to me because of the kind of importance of it for transcription and splicing dynamics. I tried to solve it, and I've spent a lot of time coming up with fancy math methods for solving these things, but none of the ones I knew how to do worked. So that made me kind of sad. But fortunately, I did come up with something that did. And here's the big picture of what did work. You take the problem that you want to solve, so it involves gene switching on the one hand, and mRNA production and degradation and whatever else on the other hand. And what you do is you say, instead of thinking about this as one problem, I'm going to think about this as two separate problems glued together in some appropriate way. So first step, I take a look at each individual problem, birth, death with no gene switching, and then gene switching with no mRNA. And then I solve them in a very specific way involving something I'm calling ladder operators, which I kind of came up with in this context. Then I take the combined problem and I represent that combined problem in terms of these convenient operators. And if you do that, it turns out to be something that you can solve by essentially drawing a bunch of pictures. And then you get a solution, which is nice. You can approximate it in various ways and optimize it so it runs really, really fast. That's what we're trying to do. So forget about the gene switching for now. Just imagine RNA that gets produced and degrades. So there's a picture of what the single cell trajectory might look like, and the chemical master equation we're trying to solve is the one on the bottom left. This one is pretty easy to solve, relatively speaking. I mean, it's not necessarily that easy, but easier than all the other ones. But anyway, our first step is to do a trick that's very common in math. So you have some problem that you want to solve. I want to find P as a function of time or PSS or whatever. I have this thing that I want to solve and I say, this looks too hard. I'm going to take this and I'm going to kind of rotate it, look at it in a different way, reframe the problem. And maybe when I reframe the problem, it'll turn out to be easier to actually solve. And so instead of considering the probability distribution, we're going to consider something more abstract called the generating function, which lives in this abstract Hilbert space. And it turns out that instead of the CME, now we have to solve something which looks a lot like the Schrodinger equation from quantum mechanics. And then this operator that controls the dynamics in that case, which I'm calling the Hamiltonian by analogy, it turns out you can write it in terms of some convenient operators for chemical kinetics. Anyway, this is kind of maybe too much detail even, but again, we can solve this in many ways. Here's the way that we're trying to solve it. We're seeking two operators, ladder operators, A and A plus, for which the three following things are true. One is we can write the Hamiltonian as A plus times A times a constant. Two is that A and A plus have a certain relationship, that A times A plus minus A plus times A is equal to the identity operator. And three, that A plus and A are Hermitian conjugates of each other with respect to a certain inner product. Anyway, if you take care of all these technical things, you get kind of an interesting, intuitive, and beautiful picture of what's happening. It turns out that for just birth-death, if the degradation rate is gamma, the eigenvalues of this Hamiltonian operator are negative integer multiples of gamma. So there's a state with eigenvalue 0, and then gamma, and then 2 gamma, and 3 gamma, and 4 gamma, and so on. And you can combine these different eigenfunctions in order to make the full solution. And the interpretation of the ladder operators is that they can take you between the different eigenfunctions. So if I start at the 3 gamma eigenfunction and I hit it with the up ladder operator, I get the 4 gamma eigenfunction. Or if I start with the 2 gamma eigenfunction, I can hit it with the down ladder operator and get the 1 gamma eigenfunction. And for technical reasons, it turns out to be a very nice thing to have, because if you have some calculation, what it really turns into in terms of this framework is it turns into just doing some algebra. And so it's just a hop, skip, and a jump away once you define these things to fully solve the problem. Here's the solution. It's complicated. Don't worry about it too much. At steady state, it just reduces to a Poisson distribution. I should note, because they're going to appear again, the CNs, the things that appear in that infinite series, are these things called Charlier polynomials, which are a certain relative of the Hermite polynomials. You can see the first couple of them there at the bottom. They look, like x squared, x to the fourth. It looks kind of like that. And it turns out this whole ladder operator business is equivalent to doing separation of variables and finding that your eigenfunctions are that. But for various reasons, this ladder operator of viewing things turns out to be technically superior. Anyway, so that's birth-death. So like I said, we have our big problem, split it up into 2 problems. We just basically solved this one the way we want. We found the right operators. Well, let's look at pure gene switching. Pure gene switching is maybe even easier. It's 2 ODEs that are coupled. You can kind of matrix eyes it, and then you can solve it by diagonalizing that matrix H. So it's easy to solve by itself, but again, we're trying to solve it in a very particular way. In particular, we like more ladder operators. This time, the down ladder operator is B, and the up ladder operator is B plus. And so you want to be able to write H as a constant times B plus times B. And then there's these anti-commutation relations between B and B plus you want to be true. For example, you want B times B plus V plus times B to be equal to the identity matrix. And then you want them to be Hermitian conjugates with respect to some inner product. And again, anyway, forgetting about the details too much, if you take care of these technical conditions, then your view of the problem becomes this, that you have a ladder with a ground, a bottom, and then one step, and the ladder operators take you between the top and the bottom, and that's it. Very intuitive way to view the problem. So now we're done. We found the ladder operators for both individual problems. Now the question is, can we use these two things to combine the problems in a way that makes it doable? Now I'm going to get into even more technical stuff. I hope your eyes don't glaze over too much. I'll get to pictures soon enough. But the central mathematical trick is something like this. It turns out that B and B plus, which are 2 by 2 matrices, you can combine them in a certain way to get a basis for the space of all 2 by 2 matrices. And why that's helpful is that when we consider the combined problem, where how much mRNA you produce depends upon gene state. If you're in gene state 1, you produce alpha 1. If you're in gene state 2, you produce alpha 2. You have this like matrix that appears, and that matrix being there makes the whole problem complicated. But if you use this basis, you can turn it into something that's maybe more doable. That's the basic technical idea. In particular, the Hamiltonian, which I remind you is like the thing that controls the problem in this generating function approach. If you view things with this kind of language, the Hamiltonian looks like this. It looks like the Hamiltonian for gene switching by itself, plus birth-death by itself, plus some interaction that depends upon the difference in the production rates. And this is kind of an intuitive way to view the problem, because if the production rates in each gene state were equal, then the gene switching and birth-death would be completely uncoupled. And it kind of makes a nice analogy with field theory from physics. Okay, two or three more slides and it gets a little bit better. So now we have our combined problem Hamiltonian posed in terms of these ladder operators. And you can ask the question, can I find the eigenstates of this Hamiltonian? Or in particular, can I find the ground state, which has eigenvalue 0, which corresponds to the steady state probability distribution, which is the thing that we really care about. And so what you do is you say, okay, I have the eigenstates of this thing, which are related to the ladder operators, the eigenstates of that thing. What if I just glue them together? If I glue them together and then I try to act on these glued together states with the ladder operators, it's easy to see what's going to happen. Let's say that I'm in, if you look at the bottom, at position N on the A ladder and position whatever on the B ladder. If I hit it with the A up ladder operator, then I go to N plus 1, and then I'm still at the same place on the B ladder. Or if I'm at whatever position on the A ladder and I'm at position 1 on the B ladder, I hit it with the down ladder operator. Now I'm at position 0 on the B ladder. In that kind of way, you reduce all this stuff to just algebra and doing +1 and -1. So you can ask, okay, what if I glued together the steady state probability of birth death and of gene switching? Is that my new ground state? And the answer is no. You get some correction of order D, which is like the difference of the production rates. But you can adjust that a little bit to get something that's true up to order D squared. And then you can adjust that a little bit to get something that's true up to order D cubed, and so on and so on. And in that way, kind of iteratively compute an infinite series solution to this problem in terms of the eigenstates of the original two problems. And then the coefficients, you can show they satisfy a certain recurrence relation, blah, blah, blah. But the net effect is you do all this math, and then boom, here's the answer. It's an infinite series. It's in terms of the same Charlier polynomials that appeared in the solution of the birth-death process before. And then there's this like complicated looking thing on the right. So you say, great, I have a solution. We could be done, but something interesting happens. We should not just think about, okay, are we done or not, but what did we just do? What's even happening? It turns out that all the terms in this complicated looking infinite series have a very simple interpretation in terms of pictures. So in particular, here's an example of a term that appears. It's at the bottom, C3 times C4 divided by some stuff times some other stuff. It turns out this corresponds to the picture where you go first from 0 to 1, and then from 1 to 1, and then from 1 back to 0. So you're going from 0 to 0, and you're going between 0 and 1 in 3 different steps. And if you draw this picture, each edge, there's a way to assign a number to it. You multiply the numbers together, and then you get the coefficient. And so if you're calculating steady state probability, here's an example of the first couple pictures that you need to draw in order to get the entire answer. The only rule as far as drawing these pictures goes is that you're not allowed to have a horizontal line on the bottom part. You're not allowed to go from 0 to 0. Anyways, it's kind of nice. And what's even more nice is that different special cases, there's this idea in physics when you deal with Feynman-like diagrams that depending on what's happening, some diagrams may be more relevant than others. And that's also true here. So it turns out if the gene switching rates between the two genes are the same, then only zigzag looking diagrams appear. And it turns out if one switching rate is much bigger than another one, which turns out to be the case in many bursting situations, then you only really need to compute these balance beam looking diagrams. But anyway, the net effect of this is that you can get the same answers as me just by drawing pictures. That's a good thing. Maybe let me say a couple more things and we can stop for questions. So we have this answer. Does it work? Yes, we validated it numerically against stochastic simulations and finite state projection and so on. Here's a couple different situations. And then you can ask, not just does it work, but is it fast? Did we really improve the state of the art or not? For the solution I showed you with the Feynman diagrams and stuff, it's pretty fast and it's accurate, but it's not as fast as it could be. So we came up with this other approximation, which works when there's a certain separation of timescales, when the switching kind of timescale is very different than the degradation timescale. You end up with this beautiful looking formula that doesn't involve an infinite series at all or any weird looking functions. It involves the exponential of a matrix, which is kind of surprising. And what's really nice about this formula is that from like kind of a math point of view is that it corresponds to the generating function of the Charlier polynomials, which I've referenced as appearing when birth-death appears a couple times, except you take the independent variable in that and you replace it with a matrix. So kind of weird and mysterious, but very pretty. And it turns out to be very fast. Here's the red line is a comparison of that approximate solution, which is really fast, to another analytic solution that already exists that's known to be accurate, and stochastic simulations. And you can see sometimes it doesn't get things perfect. For example, in the very, very middle of the picture, the red line is not exactly on top of the black line, but it's pretty good. Tried a bunch more things. Still pretty good in almost all situations. And the kicker, the important thing, is that it's really, really, really fast. So the red dots are our approximate solution, and the black dots are another analytic solution that's been published for the system. And it's like 10 or 100 times faster than that in some situations. So it's not just analytic being faster than stochastic simulations or whatever. This analytic is way faster than another analytic. So this is like the kind of thing you dream of. And the scaling is very nice. As your system gets bigger and more complicated, that's what the x-axis means. It doesn't really take that much longer to generate predictions or fit parameters. So this is really cool. And this means that not only did I do some fancy math, but maybe it's useful. There's some other nice features of the method. I think it's more general than some other ones. If you can find the relevant ladder operators for the original two problems, you can combine whatever you want this way. You can extend this to more gene states, maybe, if you understood kind of the math of that. And there's some nice parallels with physics. For example, birth, death plus gene switching somehow mathematically corresponds to one boson coupled to one fermion, like an electron. So kind of mysterious, but interesting. Anyway, that was that. And I think now's a good time to pause.
I actually have a question, John. On around slide 50, when you were showing the comparisons of your work, were you comparing that to the real, like, experimental data, or what was the comparison here?
Sorry, I should have been more clear. Everything that I'm going to show is simulated data, just because we're still working on kind of the technical stuff around looking at real data. Like, there's all sorts of other issues that pop up.
So this might be a naive experimentalist question, but how do you know that what you're doing is right, or it translates into how like a real splicing would work?
That's a good question. On the one hand, I'll say I don't. On the other hand, I will say that this kind of model that involves bursting and production degradation stuff, this is something that people have started to look at and compare with real data. And it seems to match reasonably well. So there's no guarantee that it will work really well, but the experimental stuff so far says it's a reasonable thing to try. Kind of the math and the biology intuition says the models look reasonable. So yeah.
Cool, thanks. We have a question from Jason Hughes. Can you do couplings of 3 types of interactions: birth, death, gene switching, and mono/biomolecular reactions?
Yeah, and I'm going to talk about that in just a second.
All right, follow-up question. Oh wait, let's see. Can this allow you to generate these formulas for an arbitrary number of gene states or birth-death states?
Yes, and I'm going to talk about that in just a second.
And if we have no further questions, we can go for it.
Cool.
I'm just going to ask a quick, very basic question to help me understand the difference between gene switching and your synthesis, the birth-death process. The gene switch, I mean, I'm trying to think about this at the level of a promoter driving the expression. I'm trying to figure out what the difference is between. So is it essentially like a buildup of enough energy that then you get any synthesis to occur? Is that the gene switching process? How do you distinguish? I'm trying to figure out what's the distinction between gene switching and the birth-death process itself.
Okay, I have to be a little bit careful here because my knowledge of real biology is lacking. But essentially, a gene is not producing RNA all the time. There needs to be like things attached and other checks that it's gone through to make sure things are okay. And so I think maybe we should think about it as that kind of thing. Like, is the polymerase attached or whatever? Is everything ready? And if it's ready, maybe it's in the on state. If stuff still needs to be recruited or whatever, maybe it's in the off state.
Yeah, so okay, that is matching my understanding or interpretation. So essentially it's the assembly of the transcription factors at the promoter that make it sufficient to then drive transcription. And then the rate at which transcription is driven, the birth-death process happens after that has been achieved. So they are two. Okay, I think I got that. Thanks.
That's a good question. All right, I guess I'll move on. Hold up. Cool. Yeah, so Jason's question was a great question. And I think I should pause and say that that's kind of one of the really nice strengths of doing math and writing down formulas. Imagine that I want to simulate a system with, I don't know, an arbitrary number of species. On a computer, you can't do that. You have to say, okay, let me simulate 10, and then simulate 11, and then simulate 12. And then maybe I can have some sort of line that predicts what will happen if I tried 35 different chemical species in the same model, something like that. But with math, you can write down a solution that works arbitrarily. And so in particular, one kind of amazing extension of what I just talked about is that you can use exactly the same math with almost no additional effort to describe a situation where you have gene switching between on and off or whatever, production of RNA in the different gene states, but then up to n, some arbitrary number n, splicing steps. So first you have unspliced, and then it gets one intron removed, and then next intron removed, and then the next intron removed, and so on. And then the end product can degrade. So this problem is way harder naively than that other one, because now you have n+1 chemical species, where n is the number of splicing steps in your model. And the chemical master equation in the bottom left, it's super complicated looking. You know, like you have to be careful when you try to confront something like this, or you're going to get hurt. But the amazing thing is that, as I said, you can follow exactly the same strategies before to get an answer. This time, you need a little bit more ladder operators. So it turns out, because you have n+1 chemical species, unspliced, one intron removed, two introns removed, and so on, you end up with n+1 ladders plus the ladder associated with gene switching. And so you just need to find a couple more ladder operators, and then you can do exactly the same thing as before. The switching ladder operator we already found. That's the same as earlier, the B and B plus. And then we just need to find A's. This time there's A0, A1, A2, whatever. As an example, when you have one splicing step, just unspliced goes to spliced, it looks like this. If you have arbitrary numbers of splicing steps, then it's more complicated. But the point, as far as the talk goes, is that it can be done. You know, don't worry about the complicated lookingness of it. You can draw the same pictures as before even, and I won't show you the details of that. The notation gets slightly more complicated because it's not that helpful. But you do that, you draw the pictures, whatever, and then here you go. Here's the solution. And the main difference between this solution and the previous one is that instead of the CNs, the Charlier polynomials that appeared before, there's these new things that are kind of a new mathematical kind of polynomial that I guess I came up with by solving this, the VNs. And that's the main qualitative difference. You just have those instead of the other things. So yeah, there you go. And then, as I said before, we came up with this special approximate formula that's really, really fast. And you can do the same thing here too. And it's got the same interpretation as related to the generating function of these special multidimensional polynomials. And the reason this is really cool is because one of the notorious problems with solving the CME is that as you get more and more numbers of species in your model, it becomes really hard to solve no matter what you do, simulations or FSP or whatever. But this formula is simple, really simple, and it runs really, really fast. And so when it's valid, this is far and away the best thing you can do currently out there. Anyway, here's an example of the kind of pictures you can get. For example, this is when there's one splicing step. The x-axis is the number of unspliced RNA there is, and the y-axis is the number of spliced RNA. It's kind of bistable. And then you can benchmark this against simulations. Gennady did this. The yellow and orange shading is the prediction, and the black on top of that is the results of Gillespie simulations. And there's some issues. Again, there's an approximation that we're doing, and we're trying to figure out where we can be confident that it works and where it doesn't. But for the most part, it works pretty well. We tried more, still works pretty well. And this picture is hilarious, and I want you to understand why it's hilarious. This is, on the x-axis, the state space size, kind of a measure of how complicated your system is. And on the y-axis, it's how long it takes to fit data, this simulated data, with this model. The red dots are fitting it with our new fast formula that I just showed you. The blue dots are how long it took to generate that data that we're trying to fit. So somehow it takes like sometimes 10 times longer to generate the data that we're trying to fit than it does to make, I don't know, 1,000 model predictions and try to fit it, which is crazy. That's like if you're multiplying 5-digit numbers and it takes you longer to write down the two numbers than it does to multiply them in your head. That's crazy. It's really fast. And that's kind of the power of this method. This is an example for one-step splicing, but it works whatever number of steps you want. Scales great, really fast. And so we need to do more benchmarking to be confident when it works and when it doesn't work, and maybe try other approximations. But we're hoping that we can extend this to cover when intron removal might not happen in a fixed order, or alternative splicing. And if we could do that, then we could really fit arbitrarily complicated realistic splicing dynamics. And then there's some kind of interesting math associated with that related to what you might call representation theory. But I'm working on that and I think it's pretty promising. So stay tuned. I think this is a really interesting avenue for combining theory, computation, and experimental data. So that's what I've done. I just have a couple more thoughts, maybe like 5 minutes or less, that I wanted to opine kind of generally about, I don't know, the scientific process and what I learned. Should I stop for questions now or should I wait until the end?
We have 5 minutes, so it's up to you. We don't have any questions in the, oh, actually we do from Leonard. Do you want me to go ahead and read that, or do you want to go through your philosophy real quick?
I'm gonna say my navel gazing stuff very quickly. Just, it shouldn't take too long. Okay. Yeah, I just wanted to quickly reflect on what I did and what I learned. There's a lot of talk now, and there will continue to be a lot of talk about the role of theory in biology. What role does it play? What does good theory look like? Is it even necessary? I'm very biased, obviously, but I think the answer is it is necessary. And you can do better work if you combine this theory kind of stuff with more typical computational approaches and experiments and all sorts of stuff. I didn't just solve this fancy math problem and then go, okay, I feel great, I solved it. It actually is useful for something in principle. And that's cool that I could help in principle analyze real data, although like I said, we're still working on that. And I don't think somebody with maybe a different background would have come up with the same ideas. So as we know, biology is very, very complicated, and quantitative biology is super complicated. And I think the kind of problems that we'll have to solve within the next couple of decades, as we really get into the nitty-gritties of all this new single-cell data that's coming out, are harder than anything we've ever had to solve in science before. Harder than sending people to the moon or understanding the standard model of particle physics or whatever. And so I think it's really going to take all kinds of people with all kinds of backgrounds. It's also very rewarding. So if you haven't sought out this kind of interdisciplinary collaboration for yourself, I'd highly recommend it. The other thing I wanted to say real quick is I've had some occasion to reflect on the life of Max Delbruck. He was originally trained as a theoretical physicist, taught by the founders of quantum mechanics in Germany in the 1920s, and became interested in problems of biology. He studied bacteriophage genetics first at Vanderbilt and then at Caltech, and ended up winning, along with making an indelible impact on molecular biology, winning a Nobel Prize in physiology and medicine. And so I kind of marveled at that. Like, how could you be so successful at both physics and biology? And I think I have kind of a simplistic version of what the answer might look like. And that's that I think he didn't care what he was called, whether he was called a physicist or a biologist or whatever. He was just somebody with basic curiosity about how nature worked. He asked questions, you know, how does nature work? How does bacteriophage genetics work? Whatever. And then, okay, how do I answer them? If his theoretical physics training and the tools he had from that weren't enough, he went and learned how to do biology experiments and stuff like that. And I think there's an important lesson. In an interdisciplinary field like this, I think it's easy to get stuck on labels. I'm a physicist, you're a biologist, you're a theorist, you're an experimentalist, and say, because I am this, this is what I do, and I don't do that. And I think it's kind of a limiting mindset. At the end of the day, we're all curious about how nature works, and we're trying to get to the bottom of it. So we ask some question about how nature works, and we try to answer it. Maybe we don't know how to answer it with the stuff we know how to do, but that's fine. We learn new stuff and try to figure it out anyway. I learned a lot of new stuff I never usually thought about while working on this. So what I wanted to leave you with is that you should not let your methods determine the questions you ask or your identity. Your questions should determine your methods. Lastly, I'm looking for jobs. So, you know, tell your friends. Thanks for listening.
This is where we would have all the applause. Great talk, John. We have one question from Leonard. Leonard Harris, and he says, when you say deriving an analytical expression for the larger problem can be done, do you mean it can be done by hand, or is it something that can be automated, say, in general purpose software?
No, you can write it down on paper. And I did write it down on paper.
Do you think it is something that could be automated though? I think that's more of his question. Is it something that has to be done by hand, or is this something that could potentially be done computationally?
So I guess there's two aspects of that. First of all, the downside of analytic stuff, of doing math, is that generally speaking, each new problem that someone gives to you, you have to start from scratch. I don't know how to automate the process of going from start to finish, of starting with the CME and then coming up with some analytical results for that. That's, who knows how to do that. But as far as the specific problems that I've talked about, and maybe related ones, once you write down the solution, you can come up with efficient ways to put them on a computer. And that's part of what I showed you. So hopefully that answered your question.
We are right at 101 actually. And we have a couple questions from Jason, but if it's okay, I would recommend maybe concluding the webinar and then switching over to the discussion, and where we can have kind of a more open conversation. How does that sound?
I'm fine
yes.
with it.
Darren?
Yep, I think we should do that. And Jason, you're welcome to join us in the follow-on chat. So.
Yeah, so I believe that Darren posted the Zoom link at the beginning of the webinar chat. So let's just hop over there and continue the conversation. And anybody's welcome to join.
Cool.
All right.
See you there.
Start up shortly. Thanks.